Hello and welcome to my poster presentation. My name is Sabine Stindt and I would like to introduce you to our project entitled IL-1 beta enhances the CXC chemokine expression of hepatitis C virus-infected cells. It is well known that HCV interferes with several intra- and intercellular signaling pathways and modulates them. It has been previously shown that the expression of the chemokines CXCL1, 2, 3, and 8 is upregulated after HCV infection. Recently, our group published an article demonstrating that this upregulation is further enhanced after EGF stimulation. As IL-1 beta is known to be upregulated in sera of HCV patients, we investigated whether this cytokine also causes an HCV-dependent upregulation of the respective chemokines. Figure 1 shows a significantly enhanced expression of all 4 chemokines in response to IL-1 beta, which is further pronounced in the presence of HCV. Experiments were performed in a model of acute HCV infection using the HCV strain JC1 and the human hepatoma cell line HCH7.5. As the HCV-mediated deregulation of the chemokine CXCL8 was already well described by our group in response to EGF, we focused on the IL-1 beta-dependent expression of this chemokine in the following experiments. The induction of CXCL8 is already detectable at the level of transcript, thus we checked for a possible change in the pre-mRNA expression in HCV-infected cells. Using an intron exon spanning primer, we could provide evidence for a regulation mechanism during the early stage of transcription, which you can see here in figure 2. It is well known that HCV interferes with the transcription factor NF-kappa B, and we also found the induction of CXCL8 by EGF to be NF-kappa B dependent. We therefore analyzed whether IL-1 beta dependent activation of NF-kappa B is also altered by the virus. First, which you can see here in figure 3, we checked for the influence of HCV on I-kappa B alpha degradation in response to IL-1 beta, as I-kappa B alpha prevents a P65P50 subunit to translocate to the nucleus by keeping it in an inhibitory complex. We found out that I-kappa B alpha degradation in response to IL-1 beta is enhanced and prolonged in the presence of HCV. In figure 4, you can additionally see that HCV causes an enhanced phosphorylation of P65. Taken together, HCV activates the NF-kappa B system in different ways in response to the cytokine IL-1 beta. Of course, now we wanted to know which effect this may have on CXCL8. So first of all, we performed a knockdown of P65 using specific siRNA and analyzing the CXCL8 transcript. In figure 5, you can see that this knockdown results in a significant reduction of CXCL8 expression in HCV JC1 infected cells. To provide further evidence for the role of NF-kappa B in IL-1 beta induced CXCL8 expression, we performed a chromatin IP, which you can find in figure 6. Binding of NF-kappa B to the CXCL8 NF-kappa B binding site was slightly enhanced in response to IL-1 beta in control cells. In the presence of the HCV replicon, we could measure a strong and significant enhancement. Finally, I would like to give you a short summary of our results. IL-1 beta induced expression of CXCL8 is significantly enhanced by HCV infection, as is the expression of the chemokine CXCL1, 2, and 3. HCV overactivates NF-kappa B P65 in response to IL-1 beta, thereby enhancing the binding of P65 to the CXCL8 promoter region, which in turn results in the upregulation of the CXCL8 transcript expression. Thank you very much for your attention and for your interest in our research.